Hello everyone and welcome to my channel. Now I'm going to present here is synthetic seed production techniques. Let's move on to the next slides. Synthetic seeds is otherwise known as the artificial seed and it was first coined by the Morashig. Synthetic seeds means artificially encapsulated plant propagation. It may be the somatic embryos, shoot beds, cell aggregates or any other tissue to form a seed as a we can use a seed as a propagations. First and foremost the things is encapsulating the somatic embryos in a protective coatings. These seeds have to have the potential to develop a whole plant in a in vitro as well as the ex vitro conditions. The concept of artificial seeds, there is a five material that included into the seed coat, which may be the gel agents, explants, usually somatic embryos, and additional materials such as antibiotic and antifungus to protect. It may be the pathogen free from plants and growth regulator and nutrient elements. The artificial endosperms means encapsulating matrix is serve as a artificial endosperm. These encapsulation matrix consists of two nutrient and growth nutrients is added to it. It may increases the efficiency of germinations and also the viability. And types of synthetic seeds, there are two types of synthetic seeds. One is desiccated and another one is hydrated. Desiccated seeds is completely removal of moisture content and which may be added to the polyox as an encapsulating material, which may be prevent from the growth of microorganism and non-toxic embryos. And it may take the process of desiccation takes 1 or 2 weeks or otherwise decreasing the humidity environment in environment, or otherwise keeping them on bench overnight to dry. However, desiccated seeds are beneficial only to the plant species where somatic embryos are tolerant to desiccation can be done by this method. Where susceptible, we can go to the hydrated synthetic seeds. Unhydrated synthetic seeds used as a hydrogel capsules can be used. They are more suitable for plant species where recalcitrant and sensitive to desiccations. The most common method is used is calcium alginate. There we use polyox. Here we use calcium alginate. Types of tissues used to prepare a synthetic seeds. There is four: somatic embryogenic tissues, and apical beds, and the protocoms. Somatic embryos produced from the somatic cells or vegetative cells, which help to produce a genetically identical plants. And somatic embryos are also best when you want to pick any specific characters inserted to the genes into the somatic cells. And embryogenic tissues means best when you want to proceed with clonal propagation and genetic transformation studies, but their maintenance is labor intensive and costliest method. And here is a axillary shoot beds and apical shoot tips. Do not which do not, this method do not possesses the root meristem, so they need to be the induced for root development after encapsulations. Their protocoms is specifically used for the orchids, and protocoms are encapsulated in the sodium alginate gel to form a synthetic seeds. This is the form of orchids released by the Australian University. First, in earlier days, we could not produce a somatic embryogenesis in orchid. Now, in late now for new technologies, that is from the protocons we can produce the synthetic seeds in orchids. And production of synthetic seeds. First one to choose the explant. From the explant, there is a callus inducing and inducing From the explant there is a callus, and from using the tissue culture technique, somatic embryos is induced in the callus. So the somatic embryos is proliferated, and it can be desiccated in using tissue culture techniques, and encapsulation is done and in vitro or transported to the field for germinations. And here is the method of making a synthetic seeds. First establishing the callus, and somatic induction of somatic in embryogenesis, maturation, encapsulation, synthetic seeds are tested and planted in a greenhouse. Here is an first callus tissues and induction of somatic embryogenesis, and somatic embryogenesis happen, and this is known as the somatic embryogenesis. That is here, and isolated the embryos mixed with 0.5% to 5% of sodium alginate solutions. From that we add go to the 30 to 100 ml of calcium nitrate solution. From encapsulated embryos we go to the uh, test tube, and here the germination is occurs can be seen, and testing of embryos, 10 plants conversions can be testing, and it can be tested by germination would be occur, and it may be planted in a pot, and it can be finally we can planted in a field for a plant. And components used, that is used for the synthetic seed, the encapsulation is done. Gelling agents we use sodium alginate, carrageenan, locust bean, gum, and gelatin agar, carboxymethyl cellulose, sodium pectate, and tragacanth gum. Mostly we use a sodium alginate can be used, and complexing agent is mostly for chloride content, like calcium, potassium chloride, ammonium chloride. Coating agent can be used in the polylysine, LVAX, Cantrez, glutaraldehyde, malic anhydrases, and sodium alginate. And growth regulator may be GA3, ABA, IAA, NAA, and zeatin kinetin. That's all. And protective chemical, and protective from chemical and microorganism is mycorrhiza. Popular with mycorrhiza and tetracycline. And tetracycline is used. That's in pathology. Um, s- Next, cefotaxime and rifampicin and activated charcoal. Here is artificial seed production and plant conservation from somatic embryos, which are the crop can be conserved, like papaya, brinjal, mango, carrot, sandalwood, brassica, alfalfa, lettuce, and celery can be uh, conversion in a somatic embryos. In axillary beds, which is eucalyptus, citrus, pineapple can be shoot tips, banana, cardamom, apple, kiwi fruit, and potato can be done. And application of synthetic seeds, and the selection of genotype and sterile and steady genotypes, uh, germplasm preservation of elite planting materials. From the synthetic seeds we can get elite planting material, and it can be preserved in the germplasm. It maintaining the genetic uniformity and varieties of crop, and producing the hybrid seed productions. Pros of synthetic seed, it may be easily handling and short and long term conservation capacity. Genetic uniformity can be maintained, and low cost quality plant. The quality can low cost, but quality plant material can be done, and allow for transportation and exchange of germplasm between the national and international laboratories, and allow economical mass production of elite plant varieties, and cost effective compared to the traditional seeds. Here we from the cons of synthetic seeds, one which may come out from it can be used as a synthetic seed process. Otherwise, that species that does not shoot in the synthetic seed productions. And the value of syn seeds are limited because of the lower survival rate of somatic embryos. The coating material used to prepare synthetic seeds should be contain nutrient supplement for the germination and growth of seeds. The concentration of these aquatic materials is considered as a limiting factor in producing the synthetic seeds. Less useful materials are available for producing the synthetic seeds, often inefficient maturation of somatic embryo, poor germination, growth, and development of plants. Okay, and then I will give you a points in synthetic seeds. There are varieties released in synthetic seeds. Varieties are released in carrot, cauliflower, and cabbages. These are all our cold crops, which may be planted in hill areas. Where from synthetic seeds there would be allied planting material and pathogen free material. There would be a nutrient for the germination and vigor and overall, and growth regulator for rooting. When we use apical bud, the rooting must not be happen. So using the growth regulator, there would be a rooting would be happened. um, and thank you for listening this lectures, and I will meet in next lectures. Until then, bye. Bye bye everyone.